Prior to meiosis, a cycle called interphase occurs. In interphase, during the G1S and G2 phases, the DNA in organelles replicate, the synthesis of proteins occurs, and the cell grows in volume in preparation for cell division. So here in prophase 1, the centrosomes replicate and begin to move to the opposite side of the cell to generate the spindle fibers, and the nuclear envelope begins to disappear. And here, the homologous pairs can be seen to line up next to each other and undergo the process of crossing over, in which an X-shaped structure is formed between the two non-sister chromatids in what is called a chiasma. Here, the exchange of similar genes or alleles occurs, which usually happens between 1 and 3 times per homologous pair. So now in metaphase 1, the nuclear envelope has now completely disappeared and the centrosomes have finished moving to the opposite poles of the cell. The homologous pairs orientate themselves along the equator of the cell, and this alignment of chromosomes is random and is known as independent assortment, which is an important factor in genetic variation. Now the spindle fiber microtubules attached to the kinetochores attach to the centromeres of the chromosomes. The anaphase 1, in which the homologous pairs are separated and pulled to the opposite ends of the cell. So in telophase 1, the chromosomes have reached the opposite ends of the cell, and the microtubules have begun to disassemble, and cytokinesis occurs. And by the end of this process, the nuclear envelope will have reformed, and the cell will be completely divided. Now in prophase 2, the nuclear envelope disintegrates, the centrosomes begin to move to the opposite sides of the cell again, and the spindle fibers begin to form. In metaphase 2, the chromosomes begin to line up along the equator of the cell. The centrosomes have now moved to the opposite poles of the cell, and the spindle fibers attach to the centromeres of the chromosomes again. In anaphase 2, the sister chromatids are pulled apart by the spindle fiber microtubules and are moved to opposite ends of the cell. And in telophase 2, a cleavage or cytokinesis occurs. The spindle fibers have now completely disintegrated and the cell completely splits, along with the reformation of the nuclear envelope. In review, we began with the diploid germ cell and went through 2 stages of division. In the first stage, the homologous pairs are split up. In the second stage, the sister chromatids are separated. And in the end, you end up with 4 haploid cells, which are gametes.